This is the Kratom Science Journal Club with Dr. Jonathan Cachet, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. Episode 9. A study on Kratom's chemical composition asks, what does the little-researched alkaloid speciofoline do? And what are the implications for drug interactions and the differences in alkaloid content in Kratom products?
It would behoove us to just mention now, for any of the podcast listeners, we're interested in hearing from y'all. If you have questions about anything related to, Kratom and Kratom science, go ahead and just tweet it at Kratom Science and Jay Cachet. If you have any other questions that have nothing to do with Kratom or science, go ahead and just shoot them over to us anyway. Yeah. We'll explore it.
Yeah, definitely. The study we're looking at today is chemical composition and biological effects of Kratom, in vitro studies with implications for efficacy and drug interactions. This was published in Scientific Reports on November 5th, so that's really recent. And this was done by Todd was the lead on that. Kellogg, I've seen his name before. I've seen it. There was one other Kratom study that they did. It was called The Chemistry of Kratom, published in July.
Mm-hmm.
And these are all people from university. A lot of people from UNC, University of North Carolina, where I used to work for 8 years. I did assistant research in the nursing school, so I sometimes put research assistant on my resume but really I was just like copying. But I learned how to use PubMed there, so that's something. But anyway,
it's all the same.
Yeah, there's Washington State, High Point University. So a team from a lot of different universities, chemistry, pharmaceutical sciences, and so what they did was, from the abstract, says here we profiled 53 commercial Kratom products using untargeted LC-MS metabolomics, revealing two distinct chemotypes that contain different levels of the alkaloid speciofoline.
That was good. That wasn't bad.
That was a good try. And I just want to tell listeners we couldn't even look this one up on Google because the alkaloid speciofoline has not been studied that much. Mitragynine and 7-hydroxymitragynine, which we're going to refer to as MG and 7-O, have been the two main alkaloids studied in Kratom. And so it says their questions they asked in doing this study are, what are the potential health implications of the variability and chemical content of Kratom preparations or products? And it says our ultimate objective with this study was to capture the variability of commercial Kratom products being employed by U.S. consumers and to evaluate the potential implications of this variability in terms of safety and efficacy. So there was a lot in this study that I didn't understand because they're getting into like molecular type of stuff, and there's a lot that, a lot of the methods and stuff that I'm just trying to read, but I'm like, okay, I don't know a lot of these words, so I'm going to rely on you, Dr. John, to explain exactly what's going on here in this study.
For sure. Yeah. We did have a lot of analytical chemistry and a lot of, I'd say intermediate to advanced statistical or data science analysis with the principal components. I just want to mention one more time real quick. So we're going to do mitragynine. We're going to say M, 7-hydroxymitragynine. We're going to say 7-O. And then for the speciofoline, we're going to say S.
Speciofoline or speciofoline?
we're very close. We're very close, no matter what. And the good thing is, there's not really anybody on the internet that could tell us that we're wrong.
Yeah, yeah. I guess the guys that did this study could probably tell us, but who knows?
Yeah, yeah,
A lot of
for
these
sure.
words are meant to be read and not spoken in normal conversation.
And I'm not sure. Did you mention? So this was published in Scientific Reports, which is a Nature publication, and the publication date is 2020. And I think it was,
November
just
5th.
Scientific Reports.
Yeah.
Yeah, there you go. November 5th, 2020. So this is hot off the presses.
Yep.
And like you mentioned at the onset, there are sort of like several different phases they went through. Ultimately, they were starting from where most kratom studies start, saying, well, there's discussion of this use of this plant and its alkaloids in the treatment of pain or as an opiate substitute, but there's lack of information known, potential interactions that they were certainly worried about in this paper. And so they decided to go on a data collection or data profiling expedition where they took 53 commercially available kratom products and then analyzed the complete chemotype of kratom alkaloids within those products. So they weren't just looking at levels of M, they weren't just looking at levels of 7O. They were looking at, I think, 20 or maybe even 30 of the kratom alkaloids commonly found in the plant. And what ended up happening, and I think we'll just hint towards it now before we get into it, but what ended up happening was that the speciofoline S ended up being the primary distinction between these two chemotype sets. Then we got into some genetics, as well as some sort of in vitro receptor binding assays, all of which were to hopefully point to the potential biological effects of differences in the chemotype.
Yeah, and so if people are listening here and you're just trying to think, how is this going to affect, my kratom consumption, or what does this mean for me? It's basically they do study that what we've talked about before, that kratom acts on opioid receptors in a way that's safer than classical opioids, number one. Number two, the question of multi-drug use. Does kratom slow down the liver enough to make it dangerous to use with other drugs?
So the idea is essentially that if people are using this kratom in a therapeutic way, then it is important for them to have, to be able to trust what the label says for metragenine content, to be able to trust that when they're buying, the same, kratom capsule or the same kratom extract from the same manufacturer, that batch to batch to batch, it's going to have the same general concentrations of alkaloids in it. And so they set out to say, okay, there's more than 40 structurally related alkaloids that could have biological activity or therapeutic value in kratom. Let's just look at 53 different commercial, retail kratom products and see how much variance is in those alkaloids. In that, too much variance could lead to different therapeutic effects. And so they wanted to see if there was broad similarities between these different commercial kratom products, or if there were differences. And so then that leads us into the two chemotypes.
Okay, and see, a lot of the people who buy kratom, they even did a test that said, is this Mitragina speciosa, or are these all actually kratom? And they came up with yes, they are. But a lot of people who buy kratom products seem to classify them into red, green, and white strains, which is not exactly, because there's only a couple different strains of trees and they don't have anything to do with the color. That has more to do with the drying process. So in general, people think red makes, is more of a mellow, white is the speedy one, and green in between. People realize when they start getting it from different vendors that is not necessarily consistent along with different batches of kratom. It's not necessarily about the color. The kratom products that they found here do vary, but how variable is that?
So they were expecting to find some like they were expecting to find more similarities between the different commercial products that they investigated, and what they ended up finding was like these two distinct groups. And so they were then concerned that maybe some of these products aren't the most classical sort of variety of the mitragynine or family, right? There's like, I think, several different types of like mitragynine blank, mitragynine, like in terms of plants. And so they were concerned, like, gosh, we found these differences here, and the differences were relatively significant. I wonder if some of these are different, subspecies within this plant family. And it ended up being that they were not. And so they essentially said, okay, well, we did find all of this chemotypic variance, a pretty substantial degree. I mean, down in table 1, they mention on how these K50, 51, 52, and 55 were different sample codes of commercial products. And there was, I think, essentially, I think it was like 4 to 10 times or fold difference, or that might have been with the receptor binding studies. But like there was a significant difference, especially when it came to the S molecule, the S alkaloid that we're talking about, to where like it was some 40x difference between the ones that had very little of it or they had a lot of it. So mitragynine content in the commercial samples varied by 4-fold, while the speciofoline content varied by more than 90-fold, with a minority of samples appearing in the high S group and then all the rest going into the low S group. But the variance there, and what you were talking about with the different colors, so they were saying they ended up being all the same genetics. So what can we attribute to causing this difference in the chemotypes and the alkaloid content? And so that's when you get into things like the growing conditions. What was the humidity? What was the temperature? What was the environment in which they were grown in like? And also a big asterisk on that potential list that could lead to this variance is different lengths and different in like sort of climatic conditions or environmental conditions when the plants, when the leaves are being dried and cured. And so from what I've read, and correct me if I'm off here, if there's something new, but like essentially the difference between the red veins and the greens and the whites is how long you let it cure or dry for, or how dry you let it get, whether it's in the sun or whether it's not. And so while I agree with you 100% that most of those colors are used for marketing purposes, not sort of scientific purposes, there can be alkaloid differences between those different colors. The thing is that I think the most important thing, which you hinted to when you were talking about it, is that essentially just because it's a red doesn't mean that every single red is going to treat or affect you the same. There's no consistency, repeatable, like over long term, repeatable between always doing red or always doing green or always doing white. So I just wanted to make sure that we hint on that sort of nuance there.
They said it's an oxindole alkaloid, whereas MG and 7O are indole alkaloids. So what's the difference between indole and oxindole?
Indole just refers to like the base skeleton structure of what the mitragynine is like built on. So while there is a chemical distinction in the actual structure of these molecules, they're still within the same family of being alkaloids from kratom. So even though there's a difference in terms of how we refer to them based on that skeletal structure, they are still alkaloids that are very similar to the structure of mitragynine or the 7O. And it's perfectly reasonable to believe or think that, well, maybe this S molecule also shows pain relieving or opiate substitute therapeutic value, just like mitragynine and the 7O. But interestingly, they found something different. Okay, so we found significant variance between all 53 of these products that we looked at. How can we separate those? Are there sort of clusters of similarities? Are there different chemotypes that sort of cluster together? And so that's the point of a principal component analysis. You're essentially looking at, for every one of these 53 commercial products, they ran it through the LCMS and they got to report back on the concentrations or the amount of 40 some kratom alkaloids in that plant, right? So you have 53 by 40 in terms of the data set that you're looking at and the concentrations there. Does that make sense? So the variables are that big, 53 by 40. And so what principal component analysis allows you to do is essentially take a very large, multivariate, complex data set and examine different combinations of those variables such that you're minimizing the differences, you're minimizing the differences or increasing the similarity by those that are close together, but maximizing it by those that are closer apart. So you're essentially trying to split it into different groups based on how similar and dissimilar all of these different points are to them. And then the other added value of PCA is that it essentially reduces the number of variables that you're looking at. Like, so it reduces the complexity and the scope and the size of the variable data set that you're looking into, without reducing or affecting the statistical significance of those differences. Yeah, so basically when they ran the PCA, they said, oh shit, this S alkaloid is what's causing, which is the primary driving factor that led us to discover these two chemotypes. We have the high S, we have the low S. And then from there, they were essentially like, well, what does this S even do?
In the one part, it says, in contrast, S does not bind at the opioid receptors and does not exhibit functional activity via the mu-opioid receptor. Further studies are warranted to characterize S and determine if the ligand may induce antinociception or other activities through non-opioid receptor systems. So does that mean they essentially tried to find out and don't know what it does?
Yeah, so I mean, you led with the conclusion there, right? So that's what they found, and it's a pretty interesting finding in and of itself, right? So they said we found all this variability. It's due to the concentrations of S. We have high S and low S. They said, well, hold on a second. Let's double check to make sure we don't have different genetics here. They did their genetic analysis. They did a blast analysis, and essentially what they found was that these are all the same species of M. spinosa, right? It's not the M. diversifolia or the other ones, right?
Yeah.
So it's all the same genetics. And then they said, okay, this is something that was true to the alkaloid content. So then they went in and said, okay, what is the potential biological activity or the implications of a high S and a low S being in the marketplace? And to examine that, they did several sort of alkaloid profile studies where they looked at, say, okay, mitragynine and 7-O bind to the opiate receptor family. They're partial agonists on those opiate receptors. Let's see if S is also that. And what they found was that not only does S not bind to the opiate receptors, it also doesn't resemble the sort of downstream cellular signaling pathways that unfold the same way that M and SO do. So, I mean, it's pretty amazing to say we don't know a lot about all of these 40 alkaloids. We know a lot about M. We know a lot about 7-O. We don't know anything about S. We can make it would be a reasonable assumption to say, well, it probably binds and hits the same receptors that M and 7-O do. Lo and behold, it does not.
So the issue of cytochrome P450, CYPs, these are liver enzymes. One of their headlines in the report was high S and low S kratom extracts have similar inhibitory effects on 3 major CYPs in vitro. So, and it also says kratom extracts and mitragynine have been tested as inhibitors of several CYPs. So what this suggests is in the liver, kratom slows down the metabolism of possibly other drugs. And so
Right.
you said you had a problem with that. So what was your issue with that?
I think the reason why at least it stood out to me was sort of how drastic they were proclaiming that this potential danger can be. And right, so I want to be careful to just to make sure like I'm not minimizing it too much.
Yeah.
But these CYP enzymes are like an enormous super family of degradation enzymes in the liver. There are hundreds, if not thousands of them. They're in almost every biological species on the planet. And so this family essentially, let's say, so if you're taking a prescription opiate or some, just any sort of drug or plant alkaloid, as it courses through your body, eventually the body has to break it down and get rid of it. So the CYP superfamily of enzymes performs this function, breaking down, foreign molecules or active molecules in your bloodstream and getting rid of them out of your body. So the risk is, and this is, theoretically the risk is, well, if you're taking something that blocks these degradation enzymes, and so they're not, removed from your body at the rate in which they normally would, you're going to have more in your bloodstream. You're going to have a higher concentration for a longer amount of time in your bloodstream, and that could potentially lead to dangerous side effects. In that, your body's just not processing it as fast as it normally would. I tried to just search for, okay, give me an example of a completely fatal drug interaction with the CYPs. Like, what's the worst? Are people like, is there ever a situation where you could take compound A and compound B, and then all of a sudden you're essentially dead? I couldn't find anything that was like explicitly, fatal interaction. And again, I don't want to minimize the drug to drug interactions here. It's something that everyone should always be aware about. But it seemed to me like the two worst examples I could find was birth control, wherein if you're inhibiting these enzymes, the birth control, the effectiveness of the birth control can be modified to a significant extent. So you could become pregnant. I would say that's, well, not fatal, certainly, certainly an unwanted outcome. The other one is with like warfarins or and other anticoagulant blood medications. So, if you are, and this generally happens as people get older in age, and they're taking these anticoagulants, the warfarins, and these blood medications. But if you're on too many anticoagulants and you get a cut, let's just say it's a minor cut, nothing bad, but your body can't start coagulating and forming a scab, it could lead to potentially dangerous effects in that you're going to lose too much blood. You're never going to heal the open wound, and that could be potentially risky. But broadly speaking, everything has interactions with the CYPs, and I know that there are probably plenty of our listeners who have read somewhere on Earwood or Reddit that if you take grapefruit juice, in addition to whatever sort of buzz you're looking for, from whatever compound, the compounds in grapefruit will block these CYP enzymes, and supposedly, quote unquote, you will increase the effects in sort of the length and the amount, or the threat, to the degree in which you feel the effects of those drugs. But even then, I would say that's probably 60% placebo effect. It's not necessarily that it is doing it at a physiological level to where you would actually necessarily notice that. So, I think I was objecting to it because of how many times it came up. It was almost on every single page. They sort of laid out this broad, super warning about we found that it had high S, and if it has high S, people are taking it and they're going to potentially die because the CYPs are getting inhibited. And it's like, well, again, let's get less doomsday about this. And it could have been mentioned, and it would have been valid to mention it, but to continually raise the alarms and tell people that it's a huge risk for adverse interactions, to the point to where maybe Kratom shouldn't even be legal, was just a little bit too far for me. And I don't know if you noticed too, but it was interesting to me how much, the degree to which they framed the use of Kratom as this like highly controversial. People report these things. So thus the controversy over the use and effectiveness of kratom was documented nearly 100 years ago.
I wrote that down because that was weird. Here's the controversy. According to Redley, mitragynine speciosa is used in Perak town against the opium habit, whilst this is 1921.
Right.
Ellen Field, a scientist. Whilst according to Dr. P.P. Laidlaw, his name's P.P. I didn't notice that one. Dr. P.P. Laidlaw, definitely 1921. Mitragynine is a local anesthetic. And their conclusion to this quote is, thus, controversy over use and effectiveness of kratom was documented nearly 100 years ago. Well, they could have been using it for both things. Well, how is that controversial? Like, it could be a local anesthetic and an opiate cessation tool.
Yeah, both things are essentially like they're like, just analogous to each other. They're not synonyms, but they're, definitely close in the thesaurus. There are differences with local anesthetics. Like Novocaine is a local anesthetic, and the mechanisms of how those work to like make your, whatever that you're rubbing it on or putting it on go numb, just localized in your mouth, for example, is much different than how opiates provide pain relief, but they're systemically distributed throughout your entire body. So you could get nitpicky about that, but to me, instead of trying to like continue to hype up this controversy and the fact that this controversy still exists today, mine would have been, thus, the use and effectiveness of kratom was documented nearly 100 years ago as something to defeat the opium habit, right? Something to help with opiate addiction. That would have been the takeaway for me. We knew that 100 years ago.
So in their discussion, it says, now, on the basis of our studies, it is reasonable to predict that individuals who self-administer kratom tea to treat pain, addiction, or depression might achieve very different results depending on the alkaloid profile of the product they use.
So that was their hypothesis, right? But then they said, in order for that to be true, the molecules that define the difference, right? So these two groups, it's S, that defines the sort of major two clusters of the chemotypes in these kratom products. So they said, well, let's see what it binds to. So in order for you to actually have and perceive differences in kratom products based on this difference in alkaloid profiles, the alkaloids would have to affect you differently. And so it's just interesting because they found that S has no activity at the opiate receptors. Now, so it doesn't mean that it's very plausible that it could be operating in other receptors and other signaling systems, but it just does not have an appreciable affinity for any of the opiate receptors. And so just on face value of that alone, if it's not modulating the opiate receptors any differently, then that's evidence that says maybe the difference in alkaloid profiles is not that significant. I think it's valuable, and this is sort of a cool study in that like they really went deep to try to figure out how S functions compared to M and 7O, but it ended up being that it's way completely different. An entirely new sort of ballpark of receptor interactions there. And so at least we can check off the list that S doesn't interact with the opiate system. Like then it's on for the next people to figure out what it actually does interact with. And then, so now we're talking about 3 of the alkaloids. Of all the 40 alkaloids, is there a whole subset that does not interact with the opiate system and it interacts with X? Or is it like just S that displays these characteristics? Pretty interesting.
Yeah, definitely. Another section was like in anti-nociception assays. 7O exhibits 40-fold greater potency than MG, 10-fold greater potency than morphine, whereas MG is less potent than morphine. Interestingly, a recent study by Krugel et al. Shows that MG can be converted into 7O both in vitro and in a mouse model. therefore, some of the in vitro activity attributed to MG may be in fact due to the action of it metabolizing it as 7O. So like they were, I read this study, and like when orally administered MG converts to 7O, doesn't it contradict the previous findings that MG is safer or less potent than 7O since it converts, it's metabolized into 7O?
So, not necessarily. Clarified a little bit, right?
Okay.
So, 7O has a much stronger binding affinity to the opiate receptors. I think that 7O is sort of several fold more potent than morphine, but then the mitragynine is not more potent than morphine. And so I think, what they're essentially describing here with the conversion is that I'm just going to make the units just unknown. So let's say you have 100 M and you have like 270 in terms of the amount that you ingested from the onset.
Mm.
What they're basically saying is that, by the time it gets to be biologically active in your system, it could be down to like 80 M and 2270. So it is true that the 7O is stronger and has a higher binding affinity and is more intense in terms of activating the opiate system. But I haven't seen any sort of evidence that suggests 50%, 70%, 80% of M is converted to 7O. It's a small percent, probably about 30, 20 or 30% that ultimately gets tripped over. And, it could lead to physiological differences. But it's unknown the rate at which that transition can occur, and if it occurs in everybody, and when it occurs in the process.
Yeah, because I was, because a lot of the times, they're saying some of these extracts contain more 7O, so therefore we should have an alkaloid profile that's exactly like the fresh leaf material, and so then it wouldn't be dangerous. But it's dangerous if there's more 7O. And kind of that Krugel study, one of the conclusions I came to was, well, maybe it doesn't matter since it's all being, some of the MG is metabolized into 7O. Is it really more dangerous to take Kratom with more 7O in it?
I would just qualify by saying is potentially more intense.
Okay.
Dangerous, I think too strong of a word. And the other thing is I'm unaware of any extract or any Kratom product that has more 7O in it than it does M. It's just the case that M is found at concentrations of hundreds, maybe 100 milligrams.
Okay.
And 7O is 2 milligrams. And so the reason why I bring this up is then if you were going to make an extract that had more 7O in it than it did M, you would literally have to put all of that processing effort and analytical chemistry effort into pulling out the M and adding in more 7O. And it's just
which I don't think any of these vendors are going to do.
No, it makes no economic sense. It makes no economic sense.
That's kind of like, don't buy marijuana. It might be laced with cocaine or something. I'm like, well, that would be like a bonus money-wise there.
Yeah.
Why would the dealer be so kind?
And thinking about the cannabinoids is a good way to sort of think about what I was just saying with the M and 7O, in that most cannabis has high levels of THC, let's say anywhere from 15 to 28% THC, and there are maybe a 0.5% of CBC. So if you wanted to make an oil or a vape cartridge that had more CBC in it than THC, you would literally have to process 100-fold of the amount of plant material, then pull out the THC and just concentrate the CBC to where you're spending all day. Let's say you burn through 100 pounds and you've got half a vape cartridge of CBC. It's just not, it doesn't make sense. for all the sort of like warning statements or like alarming statements they put in here about drug interactions or controversy related to kratom alkaloids, the one that they were like pretty straightforward and open about was the fact that metragenine and 7-O do not recruit beta-arrestin. And the fact that it doesn't recruit beta-arrestin is, and they say, an important factor separating the analgesic effect from the adverse effects of typical opiates, such as abuse liability, respiratory depression, and constipation. And so the failure to recruit beta-arrestin it essentially is a hypothesis that says there's less abuse liability, there's less respiratory depression, and there's less constipation potential, the sort of negative effects of your typical opiates in the kratom alkaloids. So they were looking at that as well as CM production. But because S does not bind to any of the opiate receptors, there's no sort of functional activity at the opiate receptors, so there would be no like opportunity to produce or not produce the downstream effect of recruiting that beta-arrestin.
Okay, and now what does beta-arrestin actually do, and does it, actually affect the respiratory system?
When an opiate binds to one of the opiate receptors, which is a G-coupled protein, it changes the like shape of that receptor. And then once that happens, there's downstream effects, and then it starts recruiting different cell surface like signaling molecules. And so arrestins are it's a type of protein that's in the cell wall that has to do a lot with regulating signal transduction at these G-coupled protein receptors. So the G-coupled protein gets activated, signal transduction starts, beta-arrestin sort of comes into the mix as well and interacts with them, and then it changes that signal transduction downstream. So, I think broadly speaking, that's what I know about the arrestin family. I don't know specifically the details about why beta-arrestin 2 is related to tolerance and abuse liability. I mean, essentially, if beta-arrestin is activated, something it could do is basically say, oh, this receptor has been activated for too long. I'm going to get a bunch more arrestins, and we're going to tag this receptor with the beta-arrestins, and then that will be a signal to the cell to pull the opiate receptor inside the cell, to take it off the cell surface. Yeah. When there's a reduction of those surface receptors, that leads to tolerance, physiological tolerance. Yeah. So if it doesn't recruit that, there's no way for the beta-arrestin to sort of then lead to the induction of the receptors. I think the take home overall on this study was there is significant alkaloid variance of the 40 kratom alkaloids in commercial products on the market. That could potentially lead to different therapeutic effects, and maybe some being positive, maybe some being negative. So they were concerned about all this variability in the alkaloid content, and they wanted to investigate what was the main reason for the two groups. They found the S molecule, and that essentially everything else after that, they were comparing M and 7-O to morphine and Damgo. Damgo is just another sort of opiate activation, or it binds with opiate receptors. So they were comparing all of these to morphine, and then they also had the S in there. And this would be the first ever published study that does a assessment of receptor binding of S at opiate receptors. And that's significant, even if the result is it doesn't bind, that is not binding is different from binding. So at least we know that now. And so it would be interesting I know at Kratom Science, you guys have that page that has all of the different alkaloids in there. Yeah. And I could just see, something interesting coming together where we're sort of taking note of these things. Like we have a chart or a table where it's like, M binds to mu, gamma, and delta, or kappa. 7-O binds to it, and then S doesn't bind to it. So it's sort of like a question mark. Well, where does it bind? What is it binding to?
Yeah.
So we're slowly ticking away and making progress in understanding the other alkaloids in the plant. I think that's probably our most frequently asked question is, tell me about some other alkaloids besides MN7O, or how that may lead to better antidepressant behavior or effects. I don't know if we're like, providing some banner, headline or that says, hey, this binded to this, and therefore this, we got a negative result, but that's still valuable to know. And so, we'll just see where we go from here as we further characterize these related kratom alkaloids.
Yeah, and so this was a pretty good study then, as far as understanding the understanding Superman.
Understanding Superman, understanding the S, and also, just acknowledging or confirming this alkaloid variance. We haven't had any evidence that this variance does lead to differential effects, but we haven't had any evidence to the opposite of that either. So at least we're digging in and making some, small incremental progress, such as the world of science.
Thanks again, Dr. Cachet. Check him out at jcachet on Twitter, ccvresearch.com. The music is Captain Big Wheel. The song is called Moonrunner. Kratom Science Journal Club is produced by me, Brian Gallagher, for kratomscience.com. Take care.